We all think we know cholesterol and vitamin D. One's the bad guy we try to keep low, the other's this sunshine vitamin. But what if I told you that deep inside our cells, they're locked in this intricate dance, and it's all controlled by a single hidden switch? We're going to dive into some amazing research that pulls back the curtain on how our bodies choose between making one or the other, a choice that has huge consequences for our health.
So let's set the stage with our two main characters. On one side, you've got cholesterol, and on the other, vitamin D. Both are, and I mean this, absolutely essential for us to live, but they play these very different and pretty famous roles in our bodies.
Cholesterol, right? It's the bedrock. It's the building block for our cell membranes, critical hormones. It's vital stuff. But we all know the story. Too much can be a big problem. And then there's vitamin D, the sunshine vitamin. You need it for strong bones, for a healthy immune system. They really seem like they operate in completely separate worlds, don't they?
Well, this is where it gets really cool. What if their production lines aren't separate at all? What if they actually share a starting point? And there's this single biological switch that decides moment to moment which one our body is going to make. That's the exact mystery we're unpacking today. Okay, so to understand this switch, we've got to meet the molecule in the middle. He's kind of the unsung hero of this whole story, a gatekeeper enzyme that stands at a major biological crossroads.
So this is it, the production line for cholesterol. Now I know, it looks super complicated, but don't worry, let's just focus way down at the end of the line. You can see how one path leads to cholesterol, but there's a fork in the road, a fork that can lead somewhere else entirely. And right at that fork, we find this molecule, 7-dehydrocholesterol. Let's just call it 7-DHC for short. Now, think of 7-DHC as a lump of raw clay. The body's holding this clay, and it has to decide what to make. Is it going to sculpt it into cholesterol, or is it going to sculpt it into vitamin D? This is where our gatekeeper comes in. There's this enzyme, DHCR7, and it has one very specific job. Grab that raw clay, that 7-DHC, and do the final step to turn it into cholesterol. It's basically the master craftsman of the cholesterol factory. But here is the first major clue in our little biological detective story. The research found that DHCR7, this super important enzyme, is surprisingly fragile. It's just naturally unstable, and it has a really short lifespan inside our cells. Now, this isn't some design flaw, no way. It turns out this weakness is the absolute key to how the whole switch works. So, if this gatekeeper enzyme is so fragile, what's controlling it? What tells it to stop working? Well, the answer is totally unexpected, but at the same time, brilliantly logical. I mean, just think about it for a second. What would be the most efficient signal to tell the cholesterol factory, hey, we've got enough, you can stop now? The study discovered that cholesterol itself is the signal. It's this perfect feedback loop. Basically, when cholesterol levels start to get high, the cholesterol molecules themselves actively tell the cell to destroy the DHCR7 enzyme even faster. And the data from the research is crystal clear on this. When scientists added extra cholesterol to cells in a lab, the activity of that gatekeeper enzyme, DHCR7, was cut in half. So the main takeaway is simple. More cholesterol means less of the enzyme that makes it. It's a beautifully simple system, really. Imagine a car factory. Once the parking lot outside is totally full of new cars, a signal gets sent back to the assembly line to shut it all down. Why make more when you've got plenty? Well, cholesterol does the exact same thing to its own production line. Okay, so high cholesterol shuts down the gatekeeper enzyme. It's a brilliant way for the body to regulate itself. But what happens to all that raw material, that 7-DHC that was supposed to become cholesterol? Well, this is where the switch flips. Right, so we've got this part down. Cholesterol levels are high, so the gatekeeper enzyme, DHCR7, its activity just takes a nosedive. The path to making more cholesterol is now getting blocked up. And here is the payoff. With the cholesterol pathway blocked, all that leftover 7-DHC starts to pile up. And our bodies are incredibly efficient. They don't let anything go to waste. So instead, that raw material gets shunted down the other path, the one that, with a little sunlight, leads to vitamin D. And production just soars. But hang on. This is the most stunning number from the whole study. When the researchers looked at the ratio of vitamin D being made compared to cholesterol, they found a 3-fold increase when that switch was flipped. 3 times. That is a massive shift in the body's priorities. Now, this whole elegant balancing act isn't just some cool piece of biochemistry, it's absolutely essential for our health. And you really start to understand how important it is when you see what happens when the switch is broken from birth.
The condition is called Smith-Lemli-Opitz syndrome, or SLOS, and it's caused by mutations in the very gene that builds our gatekeeper enzyme, DHCR7. In patients with SLOS, the enzyme barely works. This leads to a toxic buildup of the precursor, 7-DHC, and a severe shortage of cholesterol.
I mean, just look at the difference here. On the left, you can see healthy levels of the DHCR7 protein in a cell. But on the right, with some of the most common SLOS mutations, over 90% of the protein is just gone. The gatekeeper has effectively been taken out of the picture.
So this brings us to the really critical question. If the enzyme is already so unstable, and the mutation means so little of it is being made in the first place, is there anything at all that can be done to help stabilize it? To get that cholesterol factory running again, even just a little bit?
And this is where the research offers a real ray of hope. The scientists found that statins, the super common cholesterol-lowering drugs, actually helped stabilize the faulty DHCR7 protein. This treatment led to about a 3-fold improvement in the protein levels for the mutations they tested. It's not a cure, but it is a potential path forward for a devastating disease. And so we end right back where we started. It all comes down to this one incredibly clever enzyme. By understanding how this single switch works, we're not only finding potential therapies for a rare disease, but we're also getting a deeper look at the elegant balancing act our bodies perform every single second. It just makes you wonder, what other hidden switches are out there, just waiting to be discovered?